Welcome to lesson 3, structure and function of enzymes. You may well be wondering why I've gone for the structure and function of enzymes, because this is for part C of the assignment, so we're jumping a little bit ahead here. But an enzyme is a protein, so I really felt that it followed on nicely from the lesson on the structure and function of protein. The criteria on the part C that relates to enzymes is this. A series of enzyme catalyzed reactions are required for the process of cellular respiration, which obviously is the production of energy. Explain the relationship between the structure of enzymes and their function. It's again that old biological adage of looking at the structure and the function, looking at how it's built and how that suits its job. So the first thing to do really is to look at the actual function of an enzyme. What does it do? And this is one of my favourite quotes. Now it's a little bit outdated, 98. You can get a much more updated one from her more recent textbooks. But enzymes increase reaction rates by a factor of about 1 million. I just have to take a second just to take stock of just how awesome that is. So enzymes increase reaction rates in the body by a factor of 1 million. So reactions occur 1 million times faster with an enzyme than without them. So it's pretty much common sense that if we don't have enzymes, we die, basically. It's as simple as that. So we're going to look at this sort of back to front. We've talked about the function, but we really need to know about their structure and how it actually does this. How does it speed up reactions by a factor of 1 million? So a little bit about reactions to start with. We have two types of reactions in sort of biochemistry. We have catabolic reactions and we have anabolic reactions. And it's really quite straightforward. Catabolic reactions are when, let's say we have a bit of a rod of material here. Catabolic reactions are those that break a big thing down into smaller sections. Catabolic, that's when it goes from big to small. Anabolic reactions work the other way. So we'll have sort of small things here that when you join them together, when you have a reaction, you make a big thing in total. So that's going from small molecule to big. So some examples are listed underneath. So for example, the catabolic reactions, the things that break down big stuff, which tend to be the majority of enzymes in the body. They tend to break big things down into small things. So carbohydrates are broken down by amylase, sucrase, lactase and maltase. They're very, very specific shapes. As we've learned in the protein section, proteins are very specific shapes and enzymes are specific shapes. So they're really quite specific as to what molecule enzymes actually break down. Proteins are broken down by the enzymes trypsin, pepsin, peptidase, and there are others. And lipids are broken down by lipases. Now I've left this anabolic reaction blank because I kind of wondered whether you could think of anything that's been built from smaller things. What kind of reactions, what kind of molecules need to be built from smaller things? And we've come across these before. Can you think? How about RNA polymerase? The thing that makes, helps out with the creation of the mRNA strand in protein synthesis, something that we talked about in the first lesson, or even DNA synthase, when DNA replication occurs. So they're basically chaining nucleotides together to make either RNA or DNA. That's just the enzyme that speeds up the reaction. So like the first quote said, these things happen 1,000,000 times faster. So the creation of mRNA and DNA occur 1,000,000 times faster with an enzyme than without one. So without them, we would be up the creek without a paddle. So for a reaction to take place, we need some kind of activation energy. And I want you to consider these blue dots as being molecules in a container. And if we froze them in a freezer, surprise, surprise, then how much kinetic energy do you think that these molecules will have? Now, kinetic energy basically is movement. So if we froze them, how much kinetic energy would they have? Well, surprise, surprise, the answer is none or very, very little. So these molecules would be moving very slowly, if at all. So if we start to heat them up, there's my flame, do a nice red colour, amazing. If we start to heat them up, then these molecules start moving around a little bit, and we might get some kind of bump at some point, but nothing with any great power or force. Now, in order to get a chemical reaction, molecules have to smash into each other with enough force for the reaction to take place. So if we heated this up, for example, and we gave these molecules enough kinetic energy that it smashed into each other, smash, smash, Hulk smash, and all these molecules moved into each other, then we'd have the magic, we'd have the chemical reaction occurring. Now, without an enzyme, that's without an enzyme, and this is normal activation energy. This graph shows you the amount of energy that you'd have to put in to get that reaction occurring. So this is only sort of a diagrammatical form. If you were to sort of push this, if you were pushing this boulder up a hill, that from here to here is the amount of energy that you would have to put in before the ball started rolling on its own, before the actual before the chemical reaction would start. So in our instance, if we're talking about these molecules here, let's say we have to heat them up to 100 degrees to boiling point before this reaction started. Now I'm completely making this up, it's not always temperature, but we'll go with it. So that's the amount of heat in this instance, and it's only a sort of crude example that we'd have to put in order for this reaction to take place. That sets up the fundamental knowledge about what enzymes do. So let's have a look at the structure of an enzyme. This blue thing here is an enzyme, and it has a very special part called the active site. I need a different colour, so let's go light blue. This, along the black line here, is the active site of an enzyme. The active site. It's a really special part, and it has a really, really specific shape. And it's got a specific shape because an enzyme is made of protein. Now I'm hoping when I say that you're thinking about the structure of a protein. What are they made of? How are they chained together? Why do they produce a specific shape? Well, if you've been listening, they're made of amino acids that are joined together by a dehydration reaction, and they start folding up to make specific shapes. And if you look at this diagram over here, I thought it was a really nice sort of image. Forget all the numbers around it. Hopefully you can see that here you've got a helix in its secondary structure. You've got a beta pleated sheet, which is here, that flat thing there, and you've got another beta pleated sheet there, and there, and another alpha helix, and there and there. So the idea to put this big diagram in was to sort of show you that an enzyme is made of protein, and it's made of those amino acids by the peptide bonds that you've already learnt about, and it's that specific shape that gives it its special active site, and only one or a very select few molecules will fit into this active site. And the normal molecule that fits into the active site is called something called a substrate. That's the thing that fits into the active site. So here we have a bit of a process here, and we can see that we're starting with a big molecule, and we're ending up at the end over here on the right hand side with two separate molecules. So that tells me this reaction is a catabolic one. It's one where the big thing is broken into two small things, and sometimes they're broken into three things or four things, I think. So what happens is this, and we can summarize it at the bottom and see what's going on. This is what happens in an enzyme catalyzed reaction, in that the substrate binds into the active site. The enzyme changes shape as the substrate binds. We'll come to that in a second. So you hopefully can see this little bit of an area on the enzyme here. When it binds in, it actually changes shape. There is a reason for this. And then the substrate, which would be this sort of U shape plus triangle, is converted into the product. Okay, so the substrate is the thing that goes into the active site. The product or the products are the things that exit the active site. And when the substrate binds with the active site, that's called an enzyme substrate complex. So just some terms that you need to use when you are referencing this in an assignment, for example. That's generally what happens. Now, it could go the other way. It could actually go this way if it was an anabolic reaction. So these two things here, this blue thing and this triangle, would go in as substrate. They'd bind into the active site. It would change shape, and it would come out as a big thing. And that would be the way it would go for a catabolic reaction. So we call this specificity. Specificity. We call it the lock and key hypothesis, because it was when enzymes were first discovered, they knew that the active site was very, very specific shape, and that's what these proteins are. They're very, very specific, and only one or a select few things fit into the active site, rather like a lock and a key fitting together very specifically. It wouldn't be much of a lock if all different types of keys would have fit it. So that hypothesis there links to, not surprisingly, the fact that they're really, really specific. The substrate going into the active site is complementary to the way that a key fits a lock. But as was said on the 2 slides ago, the active site does change shape slightly. So although the lock and key hypothesis is true in that the active site is super, super specific, it only lets molecules with a certain shape bind into it, the active site does change shape. So if you think about a key fitting a lock, there's no change of shape there. It's a very solid structure. So although I don't want to sort of mislead you by saying the lock and key theory is not correct. It is. It explains why they're so specific. However, it doesn't explain everything, which is why scientists came up with this induced fit theory. And the induced fit theory just basically means that the active site has a slight change in shape. And hopefully you can see it here on the enzyme. So this is the substrate, or substrates going in to the active site, and hopefully you can see here, let's see if I can get green, there's the substrate, if you can see it. Oh, my black pointed to right there. It's blue on the diagram, it's hidden, but you can see this active site is completely closed up almost, and there is a reason for that, but this slide was just there to show you what the induced fit theory, I suppose, means. It means that the active site slightly changes to sort of hug the substrate, and then obviously what you'd get is the products coming off. So how does the structure of an enzyme make sure that the reactions occur 1,000,000 times faster with an enzyme than without? So this is pretty much an exact copy of the slide earlier on in the video, but we've added an enzyme. And what happens is these molecules do move around and they're attracted to the enzyme. They're attracted to the enzyme, and specifically the active site. So if I draw up here in this top right hand corner, if I draw that active site and I draw the substrate binding into it, why does that bind? Why is it attracted to this area of the enzyme? So here's the enzyme in black. Here's the substrate in blue. What is it that attracts it into that area? And you have to go back to my lessons on atoms and bonding, covalent bonding and ionic bonding. And it's the molecules, the atoms that line the substrate are attracted to the atoms or molecules on the enzyme. So they're really attracted. The electrons like to attract. You've either got electrochemical positives and negatives. It comes down to that, and it comes down to our marshmallow lesson. If you've been into it and hopefully understood it. So that's what happens. The substrate binds into that active site, and by doing so, when this shape change occurs, when the induced fit occurs on the active site, it actually almost pulls apart the bonds in the substrate. So we get kind of it provides the optimum environment for the substrates, or for the substrates to react. And that can mean, particularly when it's a catabolic reaction, making a big thing small, it can start almost tearing the atoms apart. And so it in summary, it provides the perfect environment for that reaction to take place. The result of that is shown in the graph here that I initially drew on one of the earlier slides. Now, if you imagine, or if you can remember, that what we said was, there's my phone going, we had to heat these molecules up in our first slide to 100 degrees. So that they smashed into each other with enough force that the reaction takes place. Now, if you remember, the activation energy in the first slide was way up here. It was 100 degrees. And we had to put so much energy in to get them smashing. And I hope you can see that on the graph that the activation energy required when there's an enzyme present is massively lowered. So for example, we may well only need to put 50 degrees of heat in, or we might need to heat it up to 50 degrees to get this to get this reaction rolling down the hill. So that we get the chemical reaction occurring. And the result of that is that chemical reactions occur much quicker. And they occur more efficiently because we have to put less energy in, and that is how the structure of an enzyme results in its function. So what we said in the start was that an enzyme is responsible for the speeding up of chemical reactions by 1,000,000. How does it do that? Well, enzymes are very specific in shape. They have a special part called the active site, and that's where substrates bind onto. And the fact that they provide the optimum conditions for the reaction occurring, they reduce activation energy massively, which makes sure the body's most efficient and reactions will occur quicker. This I've added in as the last slide because it sort of puts into a little bit of words or words that are similar that you will be expected to sort of build on when you write this part of the assignment. The presence of an enzyme simply makes it easier for a reaction to take place. All catalysts speed up reactions by influencing the stability of bonds in the reactants, the substrate. They may also provide an alternative reaction pathway, thus lowering the activation energy needed for reaction to take place. Okay. The only thing that I will also say to finish off is the word catalyst is something that I would expect you to know and use. A catalyst is something that speeds up, that does say speeds up a chemical reaction without being used up in the process. Now you might be thinking, what the hell does that mean? Something that speeds up a chemical reaction. I forgot to even put reaction. I can't even spell reaction. Without being used in the process. So if this enzyme here is catalyzing this reaction, then when it pops off into its sort of two parts, into its product, this enzyme does not get used up in the reaction itself, so it is free to move on and do another reaction. And that's what a catalyst is. It's not used up. It's there forever. It will not get burnt up or used in the reaction itself. It is free to go on and catalyze another triangle, and another triangle, another triangle, another triangle, until all the triangles have disappeared. And that's the meaning of a catalyst. It's something that speeds up a chemical reaction without being used in the process. So hopefully this covered everything. Let me know if you've got any questions, and good luck.